Lavinia, thank you so much. It has been such a cool day, and I really have appreciated all the presentations. So thank you to everyone. And Lavinia, thank you for hosting this event year after year and bringing in a fantastic lineup of speakers where we can all learn and hopefully expand our points of view and our frames of reference. So with that, it is my privilege to be here today to talk about bridging silos in an effort to unravel the role of infections in Alzheimer's disease. And really more particularly, today I want to focus a little bit on consensus. With that, my role is kind of interesting. I founded a global research consortium in 2017 called Intracell Research Group. And Intracell focuses on building research collaborations around the concept of microbial pathogenesis in asthma and Alzheimer's disease. And then recently, in the last year, we founded another organization, which is a collaboration with global reach, very interdisciplinary, called the Alzheimer's Pathobiome Initiative. And very pleased to say Lavinia is one of our co-founders and a fantastic contributor. But before I talk about anything related to what we're doing, I need to talk about why I'm here and why I do the work I do. So I just want to read a quote from one of my, so this quote appeared in a book. I'd never seen it before. It appeared in Siddhartha Mukherjee's Emperor of All Maladies at the front end of one of his chapters. And the book is absolutely stunning. I imagine many of the people here have read it, and it's about cancer. And in the book, he has this Susan Sontag quote, and it is that illness is the night side of life, a more onerous citizenship. Everyone who is born holds dual citizenship in the kingdom of the well and the kingdom of the sick. And although we all prefer to use only the good passport, sooner or later, each of us is obliged, at least for a spell, to identify ourselves as citizens of the other place. And this picture is actually me in 2015 during the hardest year of my life. Those are my two boys. They're 3 and 5 years old. And there aren't a lot of pictures of me from that year because when you're really chronically sick, you're missing. You're not present in your life. During that course of a year, I went from being a very healthy person other than chronic lifelong asthma, which had been previously well controlled on inhaled corticosteroids. I literally went to being severe refractory asthma, unable to breathe. I spent a period of 6 months awake every night, practically gasping for air. And I also developed during this time frame a chronic bladder pain disorder called interstitial cystitis, which was a crushing blow. It is considered to be one of the most painful and devastating disorders that a person can suffer from. People with this bladder pain disorder effectively always feel like they have a urinary tract infection, and you could visit the bathroom 70 times a day and have no relief. I suffered during this course of time. I had also developed joint pain, and then perhaps most disturbingly, neurologic symptoms that were very consistent and looked like multiple sclerosis. So during this time, it was obviously incredibly challenging. And as these different conditions sort of stacked up, I began to realize the more and more specialists I had on my team, up to about a dozen, that none of them were thinking together. None of them were thinking globally about my body as a system. What would take a young, healthy person who never, abused their body in any way, shape, or form from being healthy, able to run a half marathon, to being this sick? And each time I would posit that question to one of my specialists, whether it was a pulmonologist or a urologist, I'd say this all sort of happened at once. Is there something that could possibly underlie all of it? Why is my immune system so angry? With that, I did a Google search when I finally got so concerned that my illness would continue to progress, that I would eventually not be here to see how, my children turn out and what happens when they grow up. I did a Google search that inquired about the relationship between atypical pneumonia, which was one of my diagnoses over the course of this horrible year, and interstitial cystitis. And this Google search, I like to say, changed the whole rest of my life. So that sad part kind of is over. And I want to show you the person who I ended up finding who saved my life. So this is Charles W. Stratton. Unfortunately, the world lost him last year, but he was the director of clinical microbiology and an ID specialist, infectious disease, at Vanderbilt University Medical Center for 42 years. He was one of the smartest people I had ever met in my life, but with an uncanny ability to take what is very complex and make it simple. He had published research on a very unusual stealthy bacterium called Chlamydia pneumoniae and the relationship it may have to interstitial cystitis. They did a small study at Vanderbilt involving a urology colleague where they were able to pick up on this intracellular respiratory bacterium, not the sexually transmitted chlamydia, in the bladders of women that had this terrible, incurable bladder pain disorder. And while the cohort was small, 18 patients, the conclusion was, well, while this is still obviously an association, it's possible that organisms of this type that can reside inside human cells and induce chronic inflammation could get involved in chronic inflammatory diseases like this one. And so larger studies should be done. With this Google search that I did and information that I dug up on this individual, I must say too, I had worked for Pfizer and Genentech before becoming a mother. So I knew how to read and interpret medical literature. And I will say that that piece was instrumental in my ability to find Dr. Stratton. So I reached out because typically you can find the corresponding author's email address. And I did not expect this person to answer. I literally wrote my whole medical history in an email, including the last terrible year, and said, really interested in your work on Chlamydia pneumoniae. Could we talk about this? And the rest is history. He ended up having a conversation with me and replying within a matter of 5 minutes. I would come to learn many years later, after becoming a patient, a mentee, a friend, and a colleague, that he would hear from patients like me every single week that so happened to be able to navigate the medical literature and find this information. So Dr. Stratton treated me with a combination of antibiotics, and all of these different conditions I had developed went into remission. And so I have this quote here that speaks to the situation, and it's that I will never forget this person who brought me a torch in darkness. And the torch wasn't just treatment when you're sick or compassion when you're hurting and you're looking for solutions. It was actually the gift of the mentorship, teaching me the things that he had learned about obligate intracellular organisms and how they could potentially play a role in certain cases of chronic disease. So ever since then, now this is going on 9 years ago, I've been asking questions, right? So I have this Einstein quote here: to raise new questions, new possibilities, to regard old problems from a new angle requires creative imagination and marks real advances in science. So the question our group asks, and I'm so fortunate that, I have this incredible team of people, both at AlzPI and Intracell Research Group, that are all really passionate about the same thing. And it's what causes neurodegeneration. We want to know what upstream events can lead to the things that we observe when someone is converting from being a healthy individual to someone that might have mild cognitive impairment, and then finally progress on to Alzheimer's or other forms of dementia. So our group is focused on infection, and I need to really give context around why that is. If you take it to a really basic level, and you look at the human body, and you go to the National Library of Medicine and you look up inflammation, infection or pathogens are the first thing that appear on the list. Infection is indeed the most prominent driver of inflammation in the human body. We know that infections have the capability to fuel chronic inflammation, and chronic inflammation is observed in Alzheimer's disease as well as a whole host of other chronic diseases. We find this to be extremely compelling, and Amy Nelson mentioned it earlier, the work of Robert Moyer and colleagues. But amyloid beta has been demonstrated as an antimicrobial peptide, and we feel this is incredibly important. And we also acknowledge that the body of evidence around microbial infection and dementia and Alzheimer's has really grown over the last 30 years. Animal models demonstrate that you can inoculate animals with various heavily associated pathogens, and they will go on to develop classic AD-like pathology. We know from large biobank studies and long epidemiological research studies that viral and bacterial illnesses in general increase the risk for developing Alzheimer's. Microbes can and do infect the brain and CNS and are actually found in abundance in postmortem Alzheimer's brain tissues compared to healthy age-matched controls. We know from nationwide population-based cohort studies on viral and bacterial pathogens that they increase the risk of AD, and that targeted treatment in these two cohorts out of Taiwan actually reduced the risk if they were treated with antivirals or antibiotics. Now, of course, it's newer, but the COVID pandemic has really underscored the impact that a newer viral organism can actually have on the brain, not just in an acute sense, but long-term, with demonstration of things like brain shrinkage and atrophy, and even AD-like pathology in the brains of people that died with severe COVID. And lastly is something that I really want to focus on, and it's something called reversible dementias. So there are cases in the medical literature where infections are a primary driver of a dementia. And if they're treated with proper antimicrobial treatment, targeted treatment, they can be reversible. With that, I want to share our group's publication from June of this year in Alzheimer's and Dementia, where we effectively created a roadmap to ideally establish a consensus protocol to really interrogate the brain pathobiome in people with MCI and Alzheimer's disease. Although I want to say that this concept that we have to examine the microbes in the brain is actually applicable to things far beyond the context of Alzheimer's and dementia. But one thing that we documented that really has yet to be captured in this manner in one place was this entity of reversible dementias. So what we actually found were 22 individual case reports and 2 case series involving various forms of dementia, including Alzheimer's dementia, that were their primary driver, where the primary driver was identified in these cases as being infectious. And in each case, a lumbar puncture took place, and for whatever reason, and it varied from case to case, the clinician decided to screen for microbes in the CSF. And what was really fascinating about it was how the microbial landscape really kind of mirrored the landscape that has been demonstrated in patients with chronic meningitis. And again, this gets back to sort of bridging research silos. But effectively, bacteria, viruses, fungi, parasites, and polymicrobial infections were identified. And in the majority of the cases, if the patient was treated with a targeted antimicrobial, in many cases, they were intravenous antimicrobial drugs, antibiotics, antifungal drugs, antivirals, antiparasitics. The patients actually recovered, in some cases, to normal memory scores, which we know is a possibility from things like neurosyphilis, neuroborreliosis, which is Lyme disease. By the way, Lyme disease is one of the chronic infections that I was diagnosed with by Dr. Stratton when I was finally assessed and tested for chronic infections. So earlier today, someone raised the issue of, okay, could we use antimicrobial drugs for Alzheimer's disease? And actually, this has been tried. There have been some antimicrobial trials in Alzheimer's. However, the results have been really inconsistent. And one thing that really is a common thread between all of the trials, with the exception of really of 2 trials, is this thought about precision. So when an infectious disease specialist is encountered with a patient that has meningitis, let's say, they are going to initiate certain antimicrobial drugs empirically. But while they're doing that, they're going to get a lumbar puncture and blood cultures and start seeing which microbes are on board. And it's well documented that infections that can impact the CNS are frequently polymicrobial. So most of the work that has been done around Alzheimer's disease takes a group of people and puts them on a regimen without this concept of precision. So this was done with rifampin, an antibiotic, and doxycycline, another antibiotic. in 2 different trials. This has been done with minocycline, a tetracycline antibiotic. And currently, there is a trial ongoing using valciclovir or Valtrex, targeting herpes viruses in people that are seropositive to HSV1 or HSV2. This is happening up at Columbia with our collaborator, Dr. Devanand. And effectively, the thing that is really quite apparent when you look at those case reports is the need to really understand what's there before we treat. And also the susceptibility of the microbe to the treatment. We know that certain microbes are really difficult to eradicate, especially stealth organisms and particularly intracellulars. The other thing that needs to be considered, which was discussed earlier today, is the ability of the drug to get to the site of the infection, penetrate the blood-brain barrier, and remain there in a high enough concentration. So there are challenges, abundant challenges to this space as far as the relationship between microbial infection and Alzheimer's disease, or really anything that afflicts the brain. And some of that is actually inherent to studying the brain itself, right? It's very difficult because we can't get a brain sample in someone living to determine whether or not we find microbes there. But some of the challenges actually have to do with collaboration and what's needed in terms of generating a consensus in order to even decide how would we go about demonstrating what the brain microbiome is, or the brain pathobiome is, in someone while they're still alive and we can't access the brain. There have been really limited funding resources in this niche. Amy Nelson mentioned earlier, Dr. Leslie Norens with the Alzheimer's GermQuest. There have been some advances in the space, where a certain amount of funding has been available. The Infectious Disease Society of America has a set of grants that they give out on an annual basis now for the infectious etiology of Alzheimer's research. But largely, there's also a lack of industry involvement, and it has to do with profitability. Antimicrobial drug development is one of the arguably most struggling areas of drug development in the entire pharmaceutical industry. So there hasn't exactly been a great economic underpinning to boost this sort of workup. And then another challenge with this type of idea is how it straddles research silos, right? And even agency silos as far as funding. Is this a NIAID issue, a National Institute on Aging issue? Is it both? Whose purview is it under? And then another really big issue that we've become aware of is a lack of banked biosamples, postmortem samples that have been collected in a sterile manner, where these samples would be destined for microbiome research. We're aware of contamination being an issue. But with that, there are real opportunities to advance this concept. And so, some of the opportunities are really centered around linking together in an interdisciplinary manner to collaborate with one another to really accelerate innovation. We, as a group at Alzheimer's Pathobiome Initiative, have an ecosystem where we can share information with one another. Researchers can share samples. And by sharing samples that are a very precious resource, right, that were the result of someone losing their life, we are effectively extending what we learn from those samples. And instead of one lab analyzing them with a very narrow scope, we can truly interrogate the samples at multiple different laboratories and validate the results that one lab gets using other methodologies. We also at AlzPI are using different analytical approaches on the same data sets. We, as a part of this project, aim to really generate a consensus for detecting microbes, as well as helping to create and foster sterile autopsy protocols so that this sort of barrier with contamination isn't something that persists throughout the whole field in perpetuity. But really, by coming together, we're addressing another need, which is really raising awareness and increasing the visibility of something that may lack an economic incentive, but we can promote the fundability by working together and effectively branding what we're doing and making it widely known. So we have 3 succinct steps to our project with AlzPI. Step one has already been completed. It was our research roadmap that I showed earlier that contained those cases of reversible dementia. And in this research roadmap, we really point out the controversies and the unmet needs as far as the puzzle pieces are concerned. You know, what biosamples should we be using to detect microbes? What detection methods would be the best? We are using the template that has already been provided by Dr. Michael Wilson and other experts in neuroinfectious diseases and chronic meningitis to interrogate this research question on the Alzheimer's side of things. Again, bridging those silos. And we are very excited, you know, about this roadmap step being complete. Step two is underway, and it is multi-laboratory pathogen detection. So our team of academic labs that's affiliated with AlzPI is interrogating postmortem samples in order to effectively come up with this, what we are calling the CDP, but the consensus detection approach. We will publish those results once we procure them. And then, of course, the third step is the most important one, and it's translation. We don't just aim to do really cool research, basic research that never helps to, you know, result in anything that can assist patients and their families. What we really want is for our consensus detection protocol to be something scalable and available. Because what we don't know right now are how many cases, you know, are out in the world today. We have yet to quantify how many individuals might have an infectious dementia, like the case reports that we identified. Really, ultimately, we can use the consensus detection approach in prospective cohorts so that we can identify and quantify that group. And we hope that it leads to diagnostics, preventative strategies, as well as therapeutics. But really the key is precision. And the one thing I want to really state is that we don't believe every patient is the same. And this is why we really need to start understanding these precise upstream drivers. So I have to give a shout out to my husband, who's a really patient and supportive spouse, who followed me onto a construction site really early in the morning so I could get a picture of me holding the silos. But really, our team is bridging these gaps. We're deliberately diverse. We are cross-pollinating, you know, infectious diseases, microbiology, neuroscience, psychiatry, immunology, and more. You know, people like Amy's team that are interested also in the lung, because the body is one system, right? We know that it is, and a siloed approach is really limiting in chronic diseases that are very complex. And we are really pleased to say that we're already operationalizing this collaboration. And this is just a beginning example of some preliminary data that we're generating to show, yeah, we can indeed do this. We can share, and we can, you know, be effective in this multi-laboratory technique of using different microbial detection methods on the same samples. So we have tissues that have originated at the University of Edinburgh with my co-lead author, Dr. Richard Lathe, that are leaving and for assessment at the Philadelphia College of Osteopathic Medicine, our colleague, Dr. Brian Balin, Dr. Amy Nelson at the University of South Alabama, Dr. David Corey, who's doing fungal research at Baylor, Dr. Or Shemesh at Pitt, and Dr. Garth Ehrlich at Drexel. And sequencing data already procured by Dr. Lathe is headed to Drs. Tanzi, Kumar, and Eimer up at Harvard and MGH for them to apply their bioinformatics. We aim to publish those results in 2024. So just as an overview, we really believe that infectious diseases should be a part of the differential diagnosis in patients with Alzheimer's and other dementias. We also acknowledge that collaboration among specialties is going to be absolutely essential. We are actively partnering with academic centers, clinicians, researchers, industry, advocacy, stakeholder groups, and we really want to hear from you. You know, so we encourage you to reach out. Nothing would be possible without our incredible team. This is just a small example of some of the different areas of expertise that we have and some of our core members. And then, you know, thank you to the much larger group that has been contributing not only to this manuscript, but to this research project as a whole, which, you know, really at the end of the day is patient-led, patient-centered, and precision-centric. So with that, I'll conclude and, you know, open the floor for any questions, or we can start the panel.